Reduced rates, point out a couple of other upcoming seminars. Tomorrow in IB, it's Olin Sealander, another one of the candidates for the experimental costume piece. And then on Monday there'll be another IB seminar at 4 with Doris Batroff. Who I think is the last of the candidates to come through. So please try and get to those. Wildlife on this Friday is Adina Brisman from ESPN. To make conservation easements on working landscapes. And animal behaviour lunch tomorrow, I guess, in Hillgard with Marta de Ronte Mercado from the MVZ, talking about seabird predation by great white pelicans, hunting strategies for conservation. And any other announcements or introductions?
Group is on Monday, and it's Ted Patton-Puss reviewing his adventures with the Taliban or something.
If anyone hasn't seen Working with the Taliban, come along. It's a late story.
7 on Monday.
Please come in. If you folks can just sort of squeeze up a little on the side.
Thank you. I thought you'd understand.
One chair vacant.
Well, I'm really pleased and honored to be back in the museum. I owe the museum a great deal. A great many friends and colleagues I've met through the museum. And in fact, I found a career in biology through the museum. When I came here in 1964, transferring in as a junior, I intended to go to medical school. But in my senior year, I took the natural history of the vertebrates course taught by Ned Johnson, Seth Benson, and Robert Stebbins. And that course was just an incredible experience for me. Because I discovered, and I'm actually a little embarrassed to admit this, this was the first time in my life that I discovered that learning a subject could be tremendously fun. I'd been a reasonably good student, but I'd never discovered a subject that I was just passionate about until I took that course. And through that course, I had the opportunity to work in the museum, initially a job with Ned Johnson and then with Robert Stebbins. And that got me into the museum, interacting with the faculty and the graduate students and some really wonderful undergraduates, Steve Arnold, Ted Pappenfuss, Dick Sage, Ross Keister, just a whole crew of people. It was an exciting time. And it wasn't long before I decided that I had no interest whatsoever in medical school. And that was really a life-saving experience, because I'm quite certain now, looking back, had I gone to medical school, I would have been absolutely miserable. And I'm quite certain, looking back on it now, that I would have been a miserable doctor as well. And I cannot imagine a career that has been more fun and interesting than the one I've had. So, yeah, it's hard to think. But that's 40 years ago that I first walked into the museum, so I've been involved with the museum for almost 40% of its history. I find that really shocking. So today I want to talk about how organisms deal with environmental change. And I'm going to talk about 3 examples, one from the past, one from the present, and one from the future. And I want to start with a study that I did with Peter Ward, University of Washington, looking how changes in atmospheric oxygen levels and changes in temperature might have played a role in the high background extinction rates at the end of the Permian and the slow recovery into the Triassic. So this will be an adventure in paleophysiology. Moving then fast forward 250 million years, want to switch organisms and switch time frames and look at evidence, cytological evidence, genetic evidence of changes in genetic markers in the flies of the species Drosophilus subobscura, where we're trying to look to see whether there are changes in inversion frequency that are correlated with changes in climate. So we're going to be looking at genetic changes taking place over about 25 years. And then finally, I want to switch to the future and talk about some work in progress looking at how populations of lizards might be responding to climate warming and asking the question where, at which latitudes is climate warming going to have its biggest impact. This is sort of projecting forward to the future, but for me it's actually coming back to my beginnings because I'll be analyzing some data that I gathered as a graduate student a long time ago and then trying to use this as a basis for making some predictions about how lizard populations will respond to warming. So let's start first with the paleo example. I suspect that some of you know I am not a paleontologist by any stretch of the imagination. So how did I get involved in paleophysiology? Well, actually before that, I mentioned that these topics are an eclectic set of topics and an eclectic set of organisms. But they do have one thing in common, and that's they all share an intellectual debt to a paper published by Dan Jansen in 1967, a paper called Why Mountain Passes Are Higher in the Tropics. I was introduced to this paper the year before, right after I graduated. I'd gone on an OTS course to Costa Rica. And I remember sitting in Guanacaste one very hot afternoon, listening to Jansen give a talk on why mountain passes were higher in the tropics. And I thought it was the most interesting idea I'd ever been exposed to. It was a very synthetic idea. Basically, Jansen was arguing that you should think of mountain passes as barriers to dispersal, not because of the physical height, but because of the physiological height. And so it's a physiological barrier. And he argued that mountains passes in the tropics would be more of a physiological barrier than to temperate zone animals. And that idea I found intriguing as a beginning graduate student. That idea has haunted my entire career, and I think you'll see it plays a major role in all of the topics that we'll be addressing today. Okay. So let's move then to talking about oxygen and going back to the Permian and Triassic era. How did I get involved in that? Well, a few years ago, Peter Ward, who is a colleague at the University of Washington and a real paleontologist, walked into my office one day, and he wanted to talk about data from Robert Berner, a very distinguished geochemist from Yale. Berner uses various isotopic methods to estimate the amount of oxygen in the atmosphere over geological time. Today, oxygen levels are about 21%, but Berner's analyses suggest that the amount of oxygen in the atmosphere has changed drastically. So that if you go back to the Cambrian Ordovician era, oxygen levels were substantially lower, about 15%. Plants came onto the land and you got a rise in the amount of oxygen up to about 30%. And then at the end of the Permian, you have this major drop where oxygen levels crashed to about 12 or 13% and gradually working their way up. I knew about Berner's data. There'd been a really important paper published in Nature in 1995, Jeff Graham, Robert Dudley, Carl Gans, Nancy Aguilar, looking at some of the consequences of this. So I knew about the data. But Peter wanted to talk about these crunch periods where there was low oxygen and wanted to talk about, could we find ways to understand which animals went extinct during these time periods, which ones survived. But as Peter and I were talking, my mind started to wander, as it has a very bad habit of doing. And in this particular case, it started wandering towards mountain climbing. And the reason is I have an inordinate fascination with Himalayan mountain climbers and analyzing mountaineering data. So it was only natural that I would look at these data and say, if humans had been around in the Carboniferous when oxygen levels were half again higher than what they are today, I bet humans could just zip right up Mount Everest without supplemental oxygen. But if humans had been around in the Triassic, I bet they would have a hard time climbing very high. This is just playing. It's just mental games. But when Peter left that afternoon, I found myself absolutely captivated with this idea, and I started wondering, is it possible to go from an estimate of the amount of oxygen in the atmosphere to predict how high a mountain climber could climb in that kind of an atmosphere, assuming that humans had been alive during these time periods? I had no idea whether that could be done, but my intuition said it could be done. And so I just started thinking about it and playing. And it turns out that, yes, you can make that transformation. It's fairly easy. It just involves a little algebra. But I'll show you graphically how you do that. First of all, you need to know how does the inspired partial pressure of oxygen in a climber's lung change as a function of altitude. And that's going to decline because of the change in barometric pressure. No surprise. Then you need to know what's the minimum partial pressure of oxygen, the minimum amount of oxygen in the lung that enables the climber to continue to move. If it goes below that critical level, there's insufficient oxygen for a climber. And that will be a critical number. What is that? Well, Everest is 150 meters short of 9 kilometers. Over 150 climbers have now reached the summit of Everest without supplemental oxygen. So we know that you can go at least 150 meters short of Everest. I know that at least one climber can go to 9 kilometers. I don't have time to show you the calculations how I know that, but I'm going to set 9 kilometers as the upper limit. So we'll say humans can tolerate the partial pressure of oxygen and climb no lower than this amount, which is about 45 torr. Then, if we estimate what the inspired partial pressure of oxygen was under different oxygen levels, we can then say at what altitude did that critical partial pressure occur. And so if you go to the Permian, if oxygen was 30%, as Berner predicts, I would bet that a climber could reach an altitude of 12 kilometers, about a third higher than Mount Everest. Or in the Triassic, if oxygen had dropped to about 12%, that the maximum altitude a human could go is just a little above 4 kilometers, not even the height of Mount Whitney here in California. And if you do it for the entire Phanerozoic, you can then go from percent oxygen and map that onto the maximum climbable altitude. And these gray areas indicate the periods in the Earth's history when there's been sufficient oxygen for at least a few individuals to reach the summit of Mount Everest without supplemental oxygen, ignoring the fact that humans weren't around then. Small detail. Anyway, this is just play. I was just having a ball. And because I don't do modeling, as Monty knows very well, but this is great fun to do. We actually, on a lark, sent this to the Journal of the American Medical Association, and they published it, much to my delight. So this is probably the world's first and last study of paleo-mountaineering. Anyway, so I was doing this, and it really was fun. It was just play. Suddenly I realized, this is Dan Jansen. It's not the case that mountain passes are higher in the tropics. Mountain passes are higher in the Triassic. It's not temperature, but it's oxygen. And suddenly I just realized that this had some important implications to organisms living at the end of the Permian and into the Triassic. Recall that at the end of the Permian, or at the border of the Permian and Triassic, was the largest mass extinction ever. But prior to that mass extinction, at least according to some paleontologists, the background extinction rates were already elevated before the mass extinction. And once you cross the border into the Triassic, the recovery period was prolonged. So the obvious question is this major decline in oxygen occurring at about that time, might that have played a role in the high background extinction rates and in the prolonged recovery? And there are two obvious ways in which this could happen. One is that there would be direct physiological stresses on these organisms. There was so little oxygen in the atmosphere that animals living at sea level at this time would be like living at 10,000 feet today, which is high altitude in terms of oxygen and well above the altitudinal distribution of most organisms. The second thing that would add to the physiological stress is that during the Triassic was a time of elevated temperatures relative to the present. And what that means is that oxygen supply was very low, but metabolic demand would have been very high, so that the aerobic scope for organisms to is key for aerobic animals to be able to make ATP for activity and reproduction and so on, that scope would have been greatly reduced and could have been a severe problem for many ectotherms. That was no surprise, and this had been discussed in the literature. But it should also have, by Jansonian-type arguments, should have some important biogeographic consequences. And I can show that here. Let's imagine we go back to the Permian and we have a hypothetical ectotherm that could reach an altitude of 5 kilometers at that time, but no higher because the partial pressures of oxygen would have been too low. What would have happened to the altitudinal distribution of that species, the maximum altitude, as oxygen levels dropped into the Triassic? Well, we just simply run back from the critical partial pressure, and what you see is that its maximum altitude would be changed from plus 5 kilometers to minus 5 kilometers. Wait a minute, that's 1 league under the sea. We've got a basic problem here. So what we're arguing is that as oxygen levels drop, the altitudinal distributions would have been compressed to sea level, assuming no adaptation. And we can simulate that for, rather than just for an animal that had a maximum altitude of 5 kilometers in the Permian, we can do it for a series of altitudes and just see what would have happened to their altitudinal distributions as oxygen levels drop. And even if there were an animal that were able to go to 8 kilometers in the Permian and tolerate the low partial pressures at that altitude, again would have been compressed to essentially sea level at that time. I'm going to skip that. The consequence of this, biogeographically, of an altitudinal compression would be that large fractions of the land surface would have been physiologically inhospitable to organisms. Here what I've done is to simulate in a modern day Earth's topography a Triassic atmosphere and show the parts of the land surface that very likely would be inhospitable to ectotherms. And you can see that you're losing large fractions of the land surface that are available for colonization. And we know because of species area relationships in ecology that if you reduce the land area that's habitable, you should see a reduction in the number of species that can survive there. Moreover, you're going to have fragmentation of ranges, often smaller ranges, and that can lead to a series of problems as well. And I think this gives a new perspective on what the Earth was like in the Triassic. This was a time when the continents were together, and paleontologists have often thought that this was a time where the Earth was basically a superhighway. You could move back and forth easily because the continents were all together. But Ziegler and Rowley's data using paleoaltimetry suggests, in fact, there were a lot of mountains at this time, and the physiological data suggests that these would have been impenetrable barriers to the species. So, in fact, we see that we're predicting that, or post-dicting, that Pangaea was not at all a superhighway at all. In fact, it was a terrible place to be. And adding insult to injury, Christian Cedar and colleagues have shown that in the equatorial regions, these were very arid, dry regions as well. So that would have contributed to barriers and restricted movements of species. So, in summary, what we've tried to do in this work is to argue that these drastic changes in oxygen as you go to the end of the Permian into the Triassic, coupled with rising temperatures, would have created direct hypoxic stress on organisms. At the same time, it would have had major biogeographic consequences, leading to reduced range size, increased population fragmentation. And these things together might have contributed to the high background extinction rates at the end of the Permian and the slow recovery. It must have been a physiologically very challenging time. Is this a reasonable idea? I don't know. It's an argument that's based on plausibility. There are no data on altitudinal distributions of organisms this time and never will be. And we're making a series of assumptions that if any of these assumptions are wrong, the predictions are wrong as well. But I think it does place some new perspectives on a critical time in the Earth's history. So let's move from the past now and move towards the recent present and switch organisms from thinking about organisms that are long extinct to thinking about a fruit fly, Drosophila subobscura. This is a project that I've done in collaboration with some really great colleagues from the University of Barcelona, Joan Balanya, Josep Oller, Luis Serra, and the former postdoc, George Gilchrist. And we want to look at changes in the genetic composition of flies over the last quarter of a century or so. We can do this because, think back to the 30s and 40s when Dobzhansky was working in this country. He was studying a close relative of subobscura called Drosophila pseudoobscura. That's a species that has a number of chromosome inversions. And these are something one can see psychologically. And these were the first genetic markers that evolutionary biologists had. Dobzhansky and his students and grand students went around studying chromosome inversion frequencies, looking at changes over time, seasonal, geographic, and so on. And Dobzhansky inspired a great deal of work in Europe, and much of that work was done on pseudoobscura. So in the 40s and 50s, there are very great many studies that have looked at inversion frequencies and provide a timestamp for looking at genetic changes over time. Subobscura's native range is in the Old World, predominantly in this range. It was introduced into Chile in 1978 and is now found around fairly broad latitudinal range and actually has crossed the Andes as well. Shortly thereafter colonized the western coast of North America and has now gotten as far east as Salt Lake City. Probably will be on the east coast in a few years. These flies have been studied extensively, primarily by scientists in Barcelona and some of my collaborations with them, using these invasive species as an opportunity to study rapid evolution. But we can also look at it to look at genetic changes in response to climate warming. Why? Because as has been well known for decades, if you look at the frequency of particular inversions, like the standard inversion on the E chromosome, and look at how that frequency changes with latitude, you see very striking latitudinal declines. So if you collect flies from Northern Europe, almost all of the flies have the standard inversion on the E chromosome. You go to Southern Europe, it's almost absent. So there seems to be a correlation with latitude and climate. If that is the case, then you can ask the following question. Let's imagine that this sample was taken in 1940. The high frequency of this inversion at that site. What would you expect to see if climate warming had occurred at that site? How would you expect to see the inversion frequency change? And you would expect to see it decline because of higher temperatures. That inversion would seem to be disadvantageous at high temperatures. So the Barcelona team went around going back to a number of these old sites, resampling inversions, frequencies, and looking to see if the inversions had changed. I was involved in the collections in Chile and North America. I did not score any of the inversions. They tried to teach me how to do that, and I have to say that is the single hardest thing I've ever attempted to do, and I failed totally. It's my respect for Theodosius Dobzhansky and his students, and anyone who does inversion work has gone up dramatically. Anyway, here's Joan Balanya on the island of Chiloé collecting flies. Collecting flies is not like collecting lizards. It's boring. You run around a trap line, you collect the flies, you have to go back to the hotel to find out what you have. It's pathetic. But they are good for certain evolutionary studies. Incidentally, Darwin visited this island on the Beagle and was not very fond of Chiloé and described it as a most forlorn and deserted place. And it's really sad that he didn't discover a restaurant that we did. here's George and I enjoying oysters, oysters poland. Had Darwin found this, I'm sure he would have not been quite so derogatory about his experiences. Anyway, the data set consisted of 26 localities for which we have two samples, an old one and a recent sample, and the average separation time is 24 years. So a reasonable separation time. We then, for each of those sites, computed the temperature index for the initial sample and time period and the recent time period, and we computed a PC score for genome-wide for all of the chromosomes and the inversions. What we want to see is in the time interval of the 24 years on average, have climates gotten warmer at sites, and if so, do we see a change in the inversion frequencies with a higher frequency of the putatively warm adapted genotypes? And that's in fact what you see. So here we plotted the change in temperature over the time period and the change in chromosome score. A higher score here is indicative of patterns that you see at low latitudes for flies, so presumably warm adapted. And in 22 out of 26 sites, the temperatures have gotten warmer over that time interval, and the inversion frequencies that you see are higher frequencies of the low latitude inversions. And this suggests that in fact we're seeing global genetic change tracking global warming. The year before we published this, Ari Hoffman's group had shown a similar finding for melanogaster in Australia, again seeing changes over time in the genetic constitution of those flies, and also work at Leviton and Etches on Drosophila robusta in North America. So taking advantage of these old collections, these museums of inversion frequency data, going back and resampling these same sites, and then looking to see if there are changes in the expected direction. And all of these studies are showing the same, that genetic change seems to be tracking global warming. There's some complications in the study. We don't know why a particular inversion is in high frequency at one latitude versus another. These are large segments of a chromosome, there are lots of genes, and it's quite difficult, I think, to do that. I think Hoffman's group in Australia has the best chance of figuring out what's going on in these changes because of the genetics of melanogaster are so highly developed. But in any case, I think all of these studies suggest that even over very short time periods, at least for organisms that have very rapid generation times, that we see some evidence of genetic change. And that suggests that there may be some ability to buffer changes in climate. And that's the good news. Well, depends on whether or not you like fruit flies, I guess. But what I want to do now is switch to lizards. This is where I started my career. It's actually really fun to go back, although I think the take-home message is a very depressing one. This is a project that's a work in progress, developed with Joshua Tewksbury, who's a colleague at the University of Washington. Curtis Deutsch, who was a postdoc at Washington, is now at UCLA as a climatologist. And some old friends, Lori Vitt, Paul Hertz, and Hector Alvarez. And the question we're asking is, where is climate warming going to have its biggest impact on lizard populations or ectotherms in general? Are we going to see its biggest effect at high latitudes or in the tropics? We first need to address the predicted climate patterns. A number of climatological models have been developed over the last few years. They all lead to the same kind of pattern, such as the one on the left, where an increasing score here indicates that over the next 100 years, you're going to see a bigger change in climate warming temperatures. So if you look at, say, the Western Hemisphere, what the predictions are that the higher latitudes will be the areas where climate will show the greatest changes. Much reduced changes in the tropics and also in the Southern Hemisphere, probably because all parts of the Southern Hemisphere are close to the oceans, which will serve as a buffer. But this pattern, which comes out of all of the models of big changes at mid to high latitudes, has led biologists to predict that the biggest biotic changes will also be at mid to high latitudes. For example, here's a quote from a paper of Terry Root and colleagues in Nature, for a well-known paper, predicting that the biggest changes for animals would be at mid to high latitudes. We think that's wrong. We think that the biggest changes, the worst changes, are actually going to be in the lowland tropics, where where most of the biodiversity of the world is. It's not at mid to high latitudes. This is where the great diversity is. And if you look at lizards in those areas, they show an exceptional diversity, many different kinds, families and subfamilies of lizards. These are very rich biotic areas for lizards as well as for insects and most other ectotherms. Why do we think that Root's prediction and those of others is wrong? It's because basically they're making an implicit assumption that if there's a change in ambient temperature, that's going to have an effect on fitness, a direct effect on fitness. And that, we think, is wrong because it completely ignores the physiology, the behavior, and the morphology of organisms. We've known since the time of Coles and Bogert, George Bartholomew in particular, that changes in the physical environment of organisms are often buffered by behavior and physiology. And you really can't ignore those. And I'll give you a simple example of why you need to consider physiology when you're thinking about climate warming. Let's imagine we have a species that has a temperature fitness relationship like this. Let's imagine that it's a thermoconformer. So the temperature goes up by 5 degrees, its body temperature goes up by 5 degrees. Simplest case. Let's imagine that at T times 0, it's experiencing temperatures here. And if there's climate warming, its fitness is enhanced. But if this is the starting point, and this is what happens to body temperature after climate warming, now its performance and fitness drops. So you can't simply say that a 5-degree shift in temperature is going to be good or bad for an organism without considering the physiology of the species. Moreover, you need to consider whether or not the species are thermal generalists or thermal specialists. Equivalent changes in ambient temperature will have very little effect on this generalist and have huge effects on animals that are specialized for temperature. Again, you can't just look at the change in ambient temperature. You need to bring in the physiology. You need to bring in the behavior. And this is where Jansen, again, comes back to haunt me, because in Jansen's paper, one of the predictions that he made was that if you look at tropical animals and you compare them against temperate zone animals, predicted that tropical animals would be specialized for temperature, they would have reduced acclimation capacities, and potentially that could make them more vulnerable to changes in climate. So to be able to evaluate that, we need data on how temperature affects the performance of organisms. And when I was a postdoc here, one of the projects that I worked on was to develop a computerized drag strip so that I could measure the thermal dependence of sprint speed on lizards. And here's one of Adam's favorite lizards. And so here's a racetrack with a series of photocells, chase the lizard down the drag strip. You could get a measure of acceleration and maximal speed. If you do that, this is for data for Dipsosaurus, you can easily estimate the critical thermal minimum and maximum temperatures at which locomotion is possible. You can estimate optimal temperatures, and you can see whether a species is adapted to high temperatures or low, generalized for temperature, or specialized. Since I started doing that, and Al Bennett working at the same time, doing this and often working together, this has sort of become a cottage industry in lizard physiological ecology, and there are now well over 70 species of lizards for which these data are available. If you plot those as a function of latitude, so we're looking at how critical thermal maximum, optimal temperature, and critical thermal minimum changes as a function of latitude, we see some really interesting patterns. First, that Jansen predicted that tropical animals are more specialized for temperature, that is, they have a narrower tolerance zone than do the high latitude species. It's not very conspicuous on this plot, but it's very significant. But something that pops out that we had not expected was that a number of the tropical species, tropical lowland species, are not very heat tolerant. Their critical thermal maxima are much lower than temperate zone species. Paradoxically, the least heat tolerant lizards in the world are in the neotropics. That came as a real surprise. It shouldn't, because I studied those lizards as part of my thesis, but I never put it together in a broad perspective. What's going on here? Well, you need to bring in the natural history, as Bartholomew and others reminded us for decades. And if you do that, you discover something very interesting. And actually, the idea here traces to a paper by Rodolfo Ruibal in 1961, working on Cuban anoles. What Ruibal realized was that if you go to the temperate zone and you look at lizards that occur there, you find that almost all temperate zone lizards live in open habitats where there's high levels of solar radiation, where the operative temperatures are high. These lizards are basking lizards. They thermoregulate very carefully. They're active at very high body temperatures, and they have high critical thermal maxima, high heat tolerance. If you go into the tropics and look at lizards, you see lizards that live in open habitats. They are also basking. They're exposed to high levels of solar radiation, and they have high critical thermal maxima, high body temperatures. But if you go into the tropical forests, you see that there's another group of lizards there that are active at much lower body temperatures. They're not basking, they're not thermoregulating, and their heat tolerances are low. So that's what's going on here in the tropics. You have the second kind of lizard that, as Ruibal predicted, simply would not be able to survive in a temperate zone environment, can only survive in the tropics because these are relatively buffered and stable environments. They're not very hot, but they're just warm pretty much year-round. There are, in fact, a great many lizards that occur in these habitats. These are data that Laurie Good compiled. We're looking at a series of sites in the neotropics and the number of species that live that are non-basking species versus the number that live in open habitats and basking. And in most sites, the number of non-basking species outnumber the basking ones. And if you look at differences in body temperatures of active animals, it's wildly different. These are running around at temperatures like a Spheloporus would in the Berkeley Hills at around 35 degrees. But the forest lizards are active at much lower temperatures. And this suggests the possibility that these tropical forest lizards, because they're active at low temperatures and are not heat tolerant, might be vulnerable to climate warming. And so we need to take a close-up look at those species and see if that's the case. So this takes me back to my thesis work, where in 1972 and 1973 I visited Puerto Rico and went to a place called Punta Salinas and studied a lizard called Anolis cristatellus, seen here, a really spectacular, great lizard to work with. It's very easy. And I measured the body temperature of these lizards during the course of the day. There were two habitats there, but I'm going to show you data on the forest animals. So this is how body temperature profile changes during the day. They're active from sunrise to sunset. They never bask in this forest. They're thermoconformers. Their body temperature just changes as the air temperature changes. This gray bar, which you may not be able to see, is their preferred temperatures. So given the choice of a laboratory gradient, this is what they select. And you can see that during most of the day, these lizards are able to achieve body temperatures very close to or within their preferred temperature range without doing anything at all. This is a great environment for this lizard. I went back a couple of years later and measured how temperature affected sprint speed, and then I could combine these two data sets to predict how running speed would change during the course of the day. It's very clear that at this site, it's a great place for Anolis cristatellus without doing anything at all in terms of thermoregulation. It can be active at body temperatures that enable it to run greater than 90% of its maximum speed from sunrise to sunset, doing nothing. Now let's imagine what happens if climate warming increases ambient temperature in the forest by 3 degrees. we can estimate what the body temperature would be. Now you see that during much of the day, the lizards would be exposed to temperatures higher than they prefer. And the consequences in terms of performance, you'd see a crash in terms of their ability to perform. Suggesting that with even a modest level of climate warming, we're going to see major physiological stress on these organisms. Here are data from Paul Hertz gathered on the southern side of Puerto Rico a few years later, a slightly warmer site, and again, the same pattern. That, again, this was when 1980, 83, 84, lizards should be active at temperatures that enable them to perform well, but with any level of climate warming, you'd see a major drop in performance. Is a 3-degree temperature shift a realistic change that we might expect? I think so. Here are data from El Verde in Puerto Rico, the famous El Verde field station, data from 1975 now up to 2007. These are maximum average temperature in the month of July. And the temperature shift over that time period has been almost 2.5 degrees. Not a trivial amount of change at all. It's just astonishingly how much it's warmed in that forest. And we can say, what would be the impact on the lizards that are there? In 1972, Preston Webster and I studied a close relative of Christotellus called Anolis gundlachii that occurs at this site. It is also a thermoconformer. It's active from sunrise to sunset. It never basks, never does anything except run around and do things that lizards do. And during much of the day, it was able to achieve temperatures that were within its preferred range. If we now add 2.4 degrees, or actually calculate what the body temperature would be, given the observed climate warming since then, this is what we see. Now, during much of the day, those lizards in El Verde should be experiencing temperatures that are much higher than their preferred levels, suggesting that these lizards should be physiologically stressed in 2008, right now, not 100 years from now, but already. Moreover, the temperatures in the forest should now be, they're not only too hot for Christotellus, but they should be moving into the temperature range that's favored by Anolis christotellus, which at this site was restricted to the open habitats. What we think is happening then with climate warming, just in the few years since we did this initial study, climate warming will stress gundlachii directly. It will make the forest habitat suitable for Christotellus, and Christotellus will move into the forest. So gundlachii is going to get hit by a direct physiological stress and by increased biotic interactions from a competing species that will now be able to invade the forest. So the idea that we're proposing is that the reason tropical forest lizards are in trouble is that they are not adapted to high temperatures, they are adapted to low temperatures, and they're already experiencing temperatures that are at or above their optimum levels, at least in the summer months. So any additional warming will cause direct physiological stress. Any warming will make the habitats that these lizards live in invadable by open habitat lizards, which do require higher temperatures, and that will lead to increased biotic stress. If plants are responding, as many studies suggest, that you're seeing decreases in plant productivity, such that the canopies might be opening up, that is going to elevate the operative temperatures inside the forest, exacerbating this cold problem. So we think that, in fact, tropical forest lizards are sadly in real trouble. And the bottom line is that even though the ambient temperature change is projected to be greatest at high latitudes, the studies that we've done and some additional modeling that Curtis Deutsch has done suggest that the biggest biological impacts will in fact be in the tropical forests, and that those animals are most vulnerable to climate warming. They are not tolerant of high temperatures. They have limited acclimation responses, and they're already experiencing temperatures that are moderately stressful for them. Plus, you'll add the biotic interactions. And I should say that although it's been really fun and interesting to go back and recycle data from my thesis, really fun. I feel like a good ecologist recycling my own data. But This has been a sort of a traumatic study for me to do because it's the first time in my life I've ever done a study that I hope is wrong. I hope that the predictions that we're making are wrong. Because if we're right, then because the biodiversity of ectotherms is so great in the tropical forest, and if those organisms are vulnerable to climate changes, the effects that we're going to see are going to be truly devastating. And that's why I hope we're wrong. So in summary, what I've tried to do today is to look at organismal change or how organisms respond to environmental change on different time scales, and used three very different examples, how changes in oxygen and temperature might have affected the physiology and biogeography of late Permian and Triassic organisms, looking at using cytological evidence from Drosophila, looking at changes over recent time in terms of genetic constitution of these flies, and showing really rapid responses to climate warming, and then finally coming around to where I started in biology, looking at how temperature affects lizards, and looking at how latitude, temperature might interact, and differences in which animals are most vulnerable. A number of people I'd like to acknowledge. These projects are all highly collaborative. I've had some just amazing colleagues over the years that are listed here. But again, I want to highlight Dan Jansen, because that paper, which I still think is my favorite paper that I've ever read in all of biology, contains so much richness, and the ideas of thinking of how organisms deal with their environment and thinking of it in terms of physiological interactions and physiological stresses. That paper has really, I think, influenced my entire career in ways that continue to surprise me and please me. Thank you.
Great. So thank you very much. Questions?
Your analysis focused on ectotherms, which pretty well covers terrestrial vertebrates. There are a lot of other organisms, plants, and so on out there. I'm wondering if you've been able to extrapolate some of these findings of non-ectothermic vertebrates. And related to that is, can these partial pressures of oxygen be translated into what they would be in aquatic environments? There must be some connection. And if so, the entire aquatic vertebrates would be at risk as well.
Yeah, we concentrated on ectotherms because at the end of the Permian, there were no endotherms. Although John Rubin and Willem Pollinius have proposed that at the end of the Permian, you start to see evidence of the evolution of endothermy in the mammal-like reptiles. And we think they're wrong.
I was thinking of non-vertebrates.
Yeah. So there were no endotherms. Now, in terms of aquatic systems, you're absolutely right. And for aquatic organisms, there's two problems. One is that as oxygen levels drop, the amount of oxygen in the water is going to drop naturally. But also because of ambient temperatures were increasing, water temperatures are going to be higher, and the solubility of oxygen declines. So you have three things that are deleterious or stressful to aquatic organisms. Low oxygen in the atmosphere, very little oxygen in the water, in part because there's little in the atmosphere, in part because the temperatures are warmer and metabolic rates are high. So we think that if one looked at, say, the altitudinal distributions of fish or aquatic insects during the time, that they should have really been hammered. And at high elevation, it would have been extremely difficult. Insects do have some advantages because of a tracheal system, which should probably make them less vulnerable. But I think aquatic organisms would have a very hard time.
Other questions? I can't resist asking one, if I may, and that is, So you made some very strong predictions about what we should see in Puerto Rico. What's happened?
Well, we need to go back and find out.
There you go.
And that's in fact what we want to do. We want to go back to some of these same sites of El Verde, Punta Salinas. I'd really like to go back to some of the sites that Rodolfo Ruibal visited in Cuba, because he did studies like that in the late 50s. It's a long time. And so we can go back, go to those same sites, check them out on Google Earth. The forests are still there. So it'll be possible to go back to the exact same sites, resample those animals, and see whether or not, in fact, temperatures have increased. Now, in the case of El Verde forest, where the temperatures have gone up by almost 2.5 degrees in 25 years, there we're making an explicit prediction not only that the body temperatures of Gundlaki are going to be substantially higher than when Preston and I were there in 72, but we're also predicting that we'll start to see incursions by Christotellus into the forest. And one of Jonathan Bloss's students this last summer was working in El Verde and found a group of Christotellus in the forest for the first time. So it's entirely possible that they're starting to move in. Whether that's a general thing or not, we don't know. But that's the prediction. Okay.
You say much about acclimation ability of tropical lizards, likely is low acclimation. But do you think there's a possibility of evolution for acclimation? What will be over the time span? If you add some selection plus an increase in acclimation.
So, first of all, about acclimation, Jansen predicted that you would see limited acclimation responses or capacities for tropical animals. Surprisingly little work has been done along that line. The best study was done by Joyce Tsuji working on Scalopris lizards, and she showed no acclimation responsive metabolism to seasonal variation in temperature. And a few other studies. Peter's done some, Bradstrom done on frogs as well. But there's a limited amount of data to suggest that tropical animals, at least lowland tropical animals, do not show the same kinds of magnitudes of acclimation responses we see in temperate zone animals. But there's not much data there. Could they show an evolutionary response? Possibly. And certainly the fly data suggests that you have something that has a very short generation time, they have some chance of tracking. Can these lizards? My suspicion is not. But no one has ever measured the heritability of any thermal trait in any lizard. We don't have any idea what the genetic heritability of these traits are. It's never been done. And if you look at comparative data and just say what kinds of variation do you see among closely related species, the general rule is that there's very little variation species to species within a genus in most cases. Thermal sensitivity seems to evolve slowly in most groups. That's very indirect data. But I think my concern is that lizards will not be able to keep up with the rates of climate warming that we're seeing in places like Puerto Rico. Not only because of the change in temperature is rapid, but because you're going to have this interaction from species coming in from the outside, adding a second stress on them. And maybe they can deal with it, but my suspicion is not. It's just purely intuitive. But and again, I hope I'm wrong. I hope they do have the capacity to deal with it. But my suspicion is that they won't.
With a reduction in can forest
Q-gens in terms of metabolic rate?
are there that's on the first
It might improve their aerobic capacity. I don't think it's going to deal with heat stress per se. And I was just trying to think, if there are data where people have looked at Q10s of tropical and temperate zone lizards, closely related species, and looked to see whether there is any difference. Right offhand, I don't recall if anyone's done that.
There are any neutral studies
For tropical and temperate? Yeah, that's what. Done that.
Okay, we're up past 1 o'clock, and some people need to go to class. If others want to stay on and chat a bit, that's fine.